Good morning, everyone. It's my honor to introduce Professor Elizabeth Head this morning as our speaker. Some background: Dr. Head obtained her bachelor's, master's, and PhD at the University of Toronto, followed by a postdoc at UC Irvine. She then accepted a position at the University of Kentucky, where she stayed until recently, but has now returned back to us in the UC system at UC Irvine, where she is currently a professor and vice chair of research in the Department of Pathology and Laboratory Medicine. Professor Head also serves as co-PI for UCI's Titan T32 grant, which provides funding and training for the translational study of Alzheimer's disease and related dementias. Dr. Head has been involved with a vast amount of research in the Alzheimer's and Down syndrome field, appearing on over 200 papers spanning the establishment of canine models, which those of us who joined in early heard some discussion and background on. For the characterization of aging, as well as the characterization of beta-amyloid plaques and other Alzheimer's and Down's related proteins at nearly every scale of life, from biochemical to behavioral science levels. So with that introduction said, I'll now hand it over to Dr. Head to present insights from studying Down syndrome as a model for the pathogenesis of Alzheimer's disease.
Great, thanks, Daniel. So when I heard this, that you guys were inviting me for a T32 activity, I was pretty excited. So I'm hoping you guys are all enjoying your T32 experience. I think they're just wonderful mechanisms for training. So I changed my title slightly. It's kind of the same topic. And what I'd like to do is go through part of the talk that I actually gave at UCSD a couple of years back before the pandemic hit. So I did this in person for the ADRC, and introduce near the end some new data and introduce you a little bit to my lab and my trainees and what they're working on. So just to hopefully inspire you to also consider maybe going into a little bit of the Down syndrome world. But I did want to start by just saying that the most important slide in this whole slide deck is the acknowledgements, and to understand that there's a huge amount of data that we work on that does span everything from molecular, genetic, all the way up to cognitive and clinical outcome measures. And it takes a village to do this work. And I'm lucky to work with two teams, one at the University of Kentucky, where I still am an active collaborator, and also here at UCI. But most importantly, a lot of this work wouldn't be possible, of course, without our participants and their families being actively engaged in this research. And they're very committed, so I'm pretty excited about that. And of course, it takes a lot of funding, and our more recent funding is a U19. And I'll tell you a little bit about the ABCDS program at the end, because the data that we're collecting on a longitudinal cohort is freely available to you upon request. So I'd like to encourage you to think about that. So I'll just kind of get us all up to speed on what causes Down syndrome and why do I think it's important to study Down syndrome in order to understand Alzheimer's disease, and what does Alzheimer's disease look like in people with Down syndrome. And then I'm going to spend the rest of the half of the talk is going more deep dive into the areas of interest that we have here in the lab. And some of these topics shouldn't surprise you, and I'm suspecting a lot of you are working on some of these areas. But we're very interested in cerebrovascular pathology in Down syndrome and its links to neuroinflammation. And I'll kind of summarize all that material and what I think the implications are in moving forward, and then a little smattering of ABCDS to give you some insights into what we're doing with this larger study. So Down syndrome, in many respects, and my colleague Juan Fortia in Barcelona, Spain, likes to say this a lot, is the most common familial form of Alzheimer's disease. In other words, there are far more people with Down syndrome who develop Alzheimer's disease than there are families with the standard genetic mutations that lead to early onset Alzheimer's disease. So our estimates are there are almost 500,000 people in the US alone who have Down syndrome. And it was originally described in 1887 by J. Langdon Down, who was a neurologist. And this is his first paper describing the phenotype of people with Down syndrome and a kind of classic picture of a baby with Down syndrome, which we are now very much more familiar with.
And this, I think we're on the wrong.
It's not advancing.
It's not advancing. Okay,
Yeah.
Let's do stop share. And I'm glad you stopped me. All right, how does that look?
Great.
That's proper slide?
Yeah.
Okay,
Yeah.
good.
We missed
All
the
right.
introduction on the history of Downs, but
There.
yeah. Okay.
Okay, good. Thank you. Even after all the Zoom, there's glitches. So this is just, I thought it's fun. I like to know a little bit about the history of these different conditions. And so here's an image of what J. Langdon Down looked like in his seminal paper in 1887, describing the phenotype of a person with Down syndrome. So what causes Down syndrome? It wasn't until 1959 that the genetic contribution to Down syndrome was identified as an extra copy of chromosome 21, which is thankfully the smallest chromosome and has the smallest number of genes. So that makes life a little bit easier for us. And it's a really interesting story on how this was discovered. And it turns out that Dr. Gautier was the one who actually visualized that extra copy of chromosome 21 in vitro, but she didn't have a microscope to take a picture of this. And she sent those images or those slides materials to Dr. Lejeune, who took a picture of this very famous image and subsequently published a paper. And he's credited with this discovery when actually, when you read the science article about this, it turns out Gautier was really the one who discovered it. So it's one of those kind of interesting historical, again, stories about Down syndrome. But it turns out there's actually 3 causes of Down syndrome. And this becomes important when you think about how we can learn a lot about Alzheimer's disease from people with Down syndrome. So the most common cause of Down syndrome is the full extra copy of chromosome 21. But there's also another cause for Down syndrome that accounts for about 3 to 4% of people, called translocation. And that means only a tiny piece of chromosome 21 gets carried on another chromosome and triplicated. So that's a natural, essentially genetic study where you have a subset of genes that are triplicated, and you can look at the phenotype of these people and see if they develop Alzheimer's disease or not, depending on which genes are overexpressed. And there's only 2 case reports of that in the literature. So they're very, very interesting people. And the third cause is mosaicism. And this is the more challenging one, but kind of interesting as well, in that not every cell in the body has those full extra 3 copies of chromosome 21, only some cells in the body. And I still don't understand if it's tissue specific or if it's distributed. We kind of measure this in blood, but you can kind of think about this as a dose effect in terms of you might have people who are 10, 20, 30% mosaicism, and you will see varying degrees of the phenotype, again, which could be super interesting for understanding Alzheimer's disease. And there's only 1 case report that really describes early onset Alzheimer's disease in a person with Down syndrome who was mosaic. And that was by John Ringman's group at UCLA. So the good news for people with Down syndrome is they're living much longer. Back in the 50s and 60s, it was very rare for a person with Down syndrome to get much out of childhood and into their teens. This has progressively improved over the years, and now we have people, certainly in our cohort, are in their early 70s. And there's a couple of reasons why we think this is happening. One is much better management of the congenital heart defects that happen when they're little, but also better management of co-occurring illnesses and access to a lifestyle that is just like everybody else, which is going to school and going to work, and in cases getting married, in some cases getting divorced. But all of these things have combined to provide people with a much longer lifespan. And not surprisingly, and just with the general population, the most rapidly rising cohort of people with Down syndrome are those over 40 years of age. So we also are essentially having a wave, a silver wave, I can't remember if that's what we call it, in that we're seeing much larger numbers of older people with Down syndrome. And that's potentially going to have a hit on our support systems, because how do we take care of people with Down syndrome when they have dementia and potentially their parents are much, much older, demented themselves, or possibly have passed away? So that's something that we'll need to think about very, very soon. So why should we study Alzheimer's in people with Down syndrome? What does this tell us about Alzheimer's disease in the general population? And I love this picture. This is Ira Lott. He's a pediatric neurologist, and it was Ira who really set me on this path along with Carl Kottman here at UCI. And he's just a wonderful clinician, very much adored by our families. But one of the key reasons for, I think, it's important to look at Alzheimer's disease in people with Down syndrome is the fact that on chromosome 21, as many of you well know, we have the amyloid precursor protein gene. We have a number of other genes that if you look at that, I'm sure you're going to find some genes relevant to your own research. One which I'll revisit at the end of the talk, S100 beta, which is a protein associated with astrocytes. But we have mitochondrial genes, we have synapse genes, oxidative stress genes, all kinds of goodies in here that might be very relevant for testing hypotheses about the role of these genes or protein pathways in Alzheimer's disease. But really, it's the amyloid precursor protein story that's carried the most weight in this area for many, many decades. And so, not surprisingly, we see a 1.5-fold overexpression, actually 1.4 to 1.5 fold overexpression of amyloid precursor protein in Down syndrome. And not surprisingly, we see increased production of beta-amyloid, which is also a consistent feature with familial Alzheimer's disease. And as you all well know, amyloid has been implicated in the development of Alzheimer's disease. Now, when I talk about amyloid and APP and Down syndrome, it does not mean I don't think other things are important, but certainly we have good evidence that APP is critically important for developing AD and Down syndrome. And if you don't have that APP overexpression, such as in a partial trisomy case, those people tend not to develop Alzheimer's disease. So it's important for this story. And when you look at the brains of people with Down syndrome across their lifespan, for example, in this series, I'm showing you from a 31-year-old, a 40-year-old, and a 54-year-old, you'll see a very systematic progressive increase in beta-amyloid deposition in the form of plaques, and this is in the frontal cortex. Indeed, when you can quantitate those plaques and plot them as a function of age, and it's not linear. It's pretty slow, gradual increase until about 40, and then it explodes in an exponential function. And not surprisingly, the tangles start to follow suit, although clearly the tangles come after the plaques, until by the age of 40, here's a standard image of the type of pathology we see in 40-year-old brains and older of significant tangles and plaques. So the useful thing about people with Down syndrome and studying their brains and thinking about AD pathogenesis is we can get a really good handle on temporal events. And here's an example of what I mean by this exponential kind of increase is when you look at the prevalence of both plaques and tangles in the brains of people with Down syndrome. This is based on some work by David Mann in 1993, and I actually took his table data and plot them instead. But when you look at this, what's really fascinating, and I'm sorry about the typo, that's 30 to 39, that when you hit around 30, things really accelerate until by the time you get to the 40s, we're maxing out on the extent of A beta and tau pathology. And so that raises some interesting questions about, well, we have an overexpression system, but we're not seeing plaques really accumulate till about 30. So something good is happening for many decades that they can turn this A beta over and it doesn't accumulate. But something breaks in around the 30s and those plaques start to hit. And then that goes for probably almost 10 years until something changes again and the tangles really start to kick in. So if we want to understand what's contributing to all these different processes, we can look at this as a function of age in people with Down syndrome, whereas we cannot do this in the general population because the age of onset of AD is quite variable. However, what's, I think, to me, extremely fascinating is that the signs of dementia in people with Down syndrome, if they do occur, don't start to really happen until about a decade after we see both plaques and tangles. So here's an example of a couple of papers that have been published. This was work done by Nicole Schopf at Columbia University, where she put together all the cohorts that had published data at the time, which was in 2002. And here's the ages of the individuals being evaluated and what % of people have signs of dementia. And again, you can see it's pretty stable until around 50. And then those numbers start to exponentially increase. What's fascinating to me, though, is this, and that is we're not at 100%. And indeed, what we're learning more and more is that percentage of people who get into their late 60s and even early 70s is starting to increase. So we used to estimate it's about 15%. It might be starting to grow. And that gives me great hope because maybe it is a matter of managing some of the risk factors like we're learning for AD in the general population. So even though we have a genetic driver, there's still something happening here that protects these folks. Then there's been a couple of studies more recently by Andre Stridham's group and Juan Fortea's group, where they looked at the age of onset of changes and kind of plotted these. And what's striking is Andre Stridham shows in the United Kingdom that around 55 is the median age where we see signs of dementia. In a completely separate cohort, Juan Fortia estimated that number to be 53. And so we're looking at our numbers in our longitudinal cohort, but our impression is it's around that same age. So that is incredibly powerful when you think about clinical trials. So when we put this together, let's start assembling a kind of a puzzle. So why don't we start thinking about an x-axis of age and when do these events happen? And that might give us an idea of maybe what leads to what. And I wouldn't go so far as to say what causes what, but certainly what events happen earlier than other events. And that becomes critically important when we think about clinical trials and what pathways are we supposed to target, because they are going to vary as a function of age. So plaques might be great to target in the 30s, but they really don't have many tangles. So maybe a tangle intervention is not critical here. In their 40s, maybe it's important to target both plaques and tangles as an intervention. So I wanted to kind of start filling in the gaps of some of this. So one of the gaps that I thought was really interesting to kind of look at in Down syndrome was cerebrovascular pathology. And there was really kind of an interesting tension on this question. And it is, when you look back at the literature for many decades, this was not an area of focus in aging and Down syndrome, and I'll tell you why that is. And this is why sometimes asking the questions that you don't expect to ask, but it gives you some really fun new leads to pursue. So people with Down syndrome typically do not develop atherosclerosis. They typically do not develop hypertension. So I think, and given their younger ages, most folks in the field thought that cerebrovascular pathology was not going to be a feature of people with Down syndrome because they don't have these vascular risk factors. However, people with Down syndrome are prone to obesity and overweight. They have sleep apnea at a much higher prevalence than in the general population. And that's primarily due to the structure of the throat and the neck. And they are overexpressing beta-amyloid. So you've got to assume there's lots of this being deposited, not only in plaques, but also in blood vessels. And that was an area that we started to kind of map down. And it was surprising there wasn't a whole lot of systematic information about this in Down syndrome. And when you look at cerebral amyloid angiopathy in the brains of people with Down syndrome, and here's just two immunohistochemistry stains, one for A-beta-42 and one for A-beta-1-40, with A-beta-1-40 being more soluble and typically more tightly coupled to CAA. We can see buckets of plaques, but you can also see these blood vessels with A-beta-42 absolutely caked on them. And when you look at A-beta-1-40, again, tons of blood vessels in the brains of people with Down syndrome have A-beta associated with them. In fact, it's just, not only is it pervasive, it's really extensive, that you just can't imagine those blood vessels are able to function properly, that they can't constrict and dilate properly. And now we have more and more evidence that that's the case. So the question we asked is, what is the extent of cerebrovascular pathology in the Down syndrome brain? Because we have these protective factors, but we also have this increased APP expression. So do they have more? Or do they have less, or do they have the same as what we might see in late onset Alzheimer's disease? And the way that we first addressed this question was by working with a wonderful biostatistician, Michael Fallon, pictured here, where we took every single ADRC case we had here at UCI with all their neuropath reports that we generate for the National Alzheimer's Coordinating Center, or NAC, and collated all those data and put in our data from our Down syndrome autopsy cases, which, by the way, are not uploaded to NAC yet, but we can talk about that later. And so we just, at first blush, just looked to see what we had that we could compare. And we focused on controls, non-demented elderly controls, late onset Alzheimer's disease, and our autopsy cases with Down syndrome. And something I really want to highlight, which is kind of a challenge throughout this research, is, of course, the brains of people with Down syndrome are much, much younger than late onset Alzheimer's disease and controls. So we always have this age issue, that it's hard to control for age across these groups. But nonetheless, we had a pretty good sample size of 32 autopsy cases with Down syndrome with vascular data on it, and specifically focused on atherosclerosis in the circle of Willis, arteriolosclerosis, and cerebral amyloid angiopathy. And we hypothesized that atherosclerosis and arteriolosclerosis would be lower in Down syndrome compared to sporadic Alzheimer's disease, but that cerebral amyloid angiopathy, in contrast, should be higher than sporadic Alzheimer's disease. And it turns out our hypothesis was correct. So when we look at these graphs, we have the severity of the pathology, so mild, moderate, and severe. The darker colors are more severe. And here's our cohorts, DS, AD, and controls. And each graph represents a different outcome measure. So atherosclerosis, not surprisingly, less frequent in brains of people with Down syndrome, more frequent in Alzheimer's disease. And in our subset anyways, we had even more frequent athero in our controls. Arteriolosclerosis, was absolutely absent in our Down syndrome cases compared to our AD and control cases. And then in contrast, cerebral amyloid angiopathy was far more extensive and far more severe in Down syndrome when we compare that to Alzheimer's disease and control cases. And then Michael kind of plotted this a different way, looking at age of people with, this is just Down syndrome, different ages of Down syndrome in our autopsy cohort. And we can see that the severity of cerebral amyloid angiopathy decreases with age. So, we have fewer cases without CAA with age, but more strikingly, there's almost a slight linear relationship of increasing severe angiopathy with increasing age. So clearly an issue for people with Down syndrome. So the next question we started to ask, and this was with a graduate student in my lab at Kentucky, Alex Hellman, was we're seeing far more bleeds. We're seeing far more cerebral amyloid angiopathy in the brains of people with Down syndrome compared to sporadic AD. One would suspect that there might be more microbleeds in the brains of these folks. So what are the consequences of this increased CAA? And so Alex started doing the study. This is back during those years when we were really having a lot of protests about standing up for science, et cetera. And Alex was very active in this movement. And what Alex did was she wanted to look at the frequency of microhemorrhages in the brains of people with Down syndrome. So she used a classic stain called Prussian blue, which identifies hemosiderin deposits. And those are deposits in the brain. Here's an example of a blood vessel. You can see this is an H&E. And here, this blue deposit is hemosiderin. Those blue deposits represent iron in the brain, and it should not be in the brain. So the only way you see this is when there's been a microbleed. What's really kind of cool about the stain, though, is that bleed could have happened decades before we looked at their brains. It doesn't tell us about when that bleed happened. Anytime you have a bleed, it just sits there forever and ever. I'm sorry, it's kind of a scary thought, but it's really useful for research purposes. So Alex did this very laborious project where she identified blood vessels and then counted microbleeds in a blinded fashion to the cases. And it was a large study of young controls who were under 40 years, middle-aged controls, 40 to 60 years. You can see here, we're trying to pick controls now that match for the typical age of our Down syndrome cases. And then we had age controls to match for sporadic AD. And in this case, we hit up the NIH Neurobiobank for younger control cases under 40 and those with AD pathology, which were typically over 40, and compared those to sporadic AD. So this is a big study, and here's some representative images. Prussian blue is not a pretty stain. When it works extremely well. So it does take a lot of manual labor to kind of look at things very carefully and quantitate them. And what Alex found was when we looked at just the presence of microbleeds in the brains of controls, we see a little bit of an increase in middle age and old controls. So I think that's actually kind of good news considering my own age. When we look at sporadic Alzheimer's disease, the average number of microbleeds in the frontal cortex is much, of course, higher. So on average, about 32 to 33 bleeds in the brain region she examined. And in people with Down syndrome who are under 40 without AD pathology, we had random infrequent bleeds. And when we look at Down syndrome with Alzheimer's disease, what's interesting to me is and look at the variability, we are not significantly different than sporadic AD. So despite all this extra CAA in Down syndrome brain, we are not seeing more bleeds than what we would typically see in sporadic Alzheimer's disease. So does that suggest to us another protective event? And it turns out infarcts, frank infarcts in people with Down syndrome is not common. So another really cool thing to look at in some more detail. However, microbleeds are important and are happening in the brains of people with Down syndrome. And a lot of the neuroimaging approaches that we're looking at now, you can clearly see these dark circles. These are microbleeds by neuroimaging, by MRI, and they tend to really favor the occipital cortex. But here is a person that came in for a standard longitudinal visit with us at the University of Kentucky. And he did his clinical evaluation, his neuropsych evaluation, and went into the scanner. And as these scans came down the pipe, we saw he was actively bleeding. So we pulled him out and sent him into the emergency room. But this is happening in people with Down syndrome, and we didn't have any clinical signs of that happening. So we can have a fun talk a little bit later about what the implications of this might be when we're thinking about, say, Aduhelm or any kind of vaccine or immunotherapy targeting A-beta in people with Down syndrome, because to me, this is a serious concern. And as I mentioned, I'm going to try and highlight some junior investigators and students in this talk. I'm really excited that Adam Brickman at Columbia University, who's been doing a lot of work on vascular pathology by neuroimaging in ADNI and other cohorts, took an interest in Down syndrome and came on board with us. And he had, Patrick Lau was his graduate student, and Patrick's thesis work was to characterize vascular pathology by neuroimaging in people with Down syndrome, and there was very little work on this. And he did this beautiful job of characterizing white matter hyperintensities, peravascular space widening, and also looking at microbleeds, as I showed you better in a different image, and frank infarcts in a cohort of people with Down syndrome. And he published one of the first papers on this. And this was really surprising to me, these white matter hyperintensities, because we typically think of those as being a consequence of hypertension, and we don't see a lot of that in Down syndrome. And certainly in our cohort in Kentucky, we didn't see a lot of this. So Adam and I have been pushing around ideas about, is this cohort dependent or is this something else? And so we have an R01 in submission, and fingers crossed we get to explore this in a little bit more detail. But Patrick did an absolutely gorgeous job on this paper. So the next question then, if we just keep kind of following the tail, and I'll be honest with you, this tail actually did not evolve in the beautiful order I'm telling you. It kind of happened all over the place. But we wanted to know, what are the consequences of all these bleeds in the brain? You would imagine there's all these proteins in the blood. They're getting into the brain. They shouldn't be in the brain. And those are probably going to be causing inflammation. So I partnered up with Donna Wilcock, who was at the University of Kentucky with me. We were delighted to recruit her a couple of years after I arrived there. And Donna and I did a lot of painting and drinking together. So what we did is I was able to provide her with frozen tissue, again for frontal cortex, and asked me why I always do frontal, where I was able to give her some young controls, middle-aged controls, and old controls, kind of the same design as I just described. And then 8 brains of people under 40 years of age without AD pathology, lots of brains with AD pathology, and some sporadic AD cases. So again, here's our ages and our postmortem intervals. You can kind of see who's our ADRC cases, which are these shorter postmortem intervals, and the NIH Neurobiobank, by the nature of how they do these collections, have longer postmortems. So another thing to keep in mind, that PMI could be potentially a covariate in these analyses. And so Donna, at that time, this was in 2000, early 2000s, so I'm sorry, it's a little bit dated, but it's surprising there's not a lot of updates on this. Used RT-PCR and a subset of markers of inflammation that she thought would be important. And the first thing she did was she looked at comparison of sporadic AD compared to their age-matched controls to understand the fold changes in RNA and these different markers of inflammation. The dotted line in each of these graphs represents no fold change. And of course, if the bar is lower, it's a decrease, and higher, it's an increase. And she clustered these genes into different classes, which we can talk about later. I know it's not a super favorite nomenclature to use, but it helped here a lot. But what we can see in sporadic AD is we see IL-6, IL-12 are increased, IL-10, and TGF-beta. So this is kind of the profile of what we would expect to see in sporadic AD. So then we just looked at the younger cases of Down syndrome compared to their younger age-matched controls. And it was remarkable to see that we already had a huge amount of neuroinflammation in these younger people. Now, of course, they're coming to autopsy for a reason, but this was a pretty consistent finding that a lot of these markers were increased, with the exception of CD86 and FCGR1 beta, similar to what we see in sporadic AD. So now what happens when you have a brain of a person with Down syndrome who died with Alzheimer's disease? It gets worse. So huge amount of increase in inflammation and a number of pro and anti-inflammatory molecules. But uniquely to Down syndrome, we see CD68 and FCGR1 beta increased. And this is not a pattern we see in sporadic AD. It's unique to Down syndrome. So the way Donna explained this to me is if you kind of break it down into these different classes, M1, M2A, M2B, M2C, you can get some idea of what the pathology underlying that profile might be. And I think really of interest to us with this M2B phenotype, it's typically associated with immune complexes forming in the brain. And that was kind of the unique pathway that came up in our Down syndrome cases. So that led us down, as you can well imagine, the next path. And that is, what do microglia look like in the brains of people with Down syndrome? And again, when you looked in the literature, there were scattered reports of people looking at microglia in Down syndrome brain with very small sample sizes, 2 or 3 cases. They tended to be end-stage cases. So far, it looked like things were the same, but there really wasn't a good systematic study of this and an understanding of how it might change with age. And so this was another study that Alex took on at University of Kentucky. And then when I moved here to UCI, I was delighted to bring Alessandra Martini on board. And Dr. Martini was able to kind of wrap up the study and offer her interpretations of what these data meant. She brought a nice background of looking at microglia in mouse models with Frank Lafurla's team. And so what we did is a case series of looking at what are the different microglial phenotypes that we observe in the brains of people with Down syndrome. And to do this study, similar type of setup, we had young controls, their age-matched cases with Down syndrome, middle-age controls, DSAD, age controls, and Alzheimer's disease. And there's our ages of death, our distribution of males and females, and our postmortem intervals. And we use the Versa Imperial scanner to scan in and digitize all the images that we had. And then each of these images was annotated. And then this is where the labor began. Every single image, the number of microglia that kind of met each of these phenotypes was counted. I'm hoping there's a better way to do this going forward because of these beautiful AI approaches we can use. But in this case, we did this manually, and actually I'll tell you about another way that it was done. So Alex and Alessandra kind of characterized these microglia by being ramified, which is what we consider the resting or surveillant microglia, whether or not they're amoeboid, which is apparently represents more of an acute inflammatory response. Hypertrophic, which we're all super familiar with. These are active and busy microglia that are trying to deal with damage in the brain. Dystrophic microglia, which are microglia that have these blebs and their processes are a little bit beaded. And Wolfgang Streit actually described this as part of the lifespan of a microglia. This is their old age senescence and dying morphology. And then we have these rod-shaped microglia, which are really cool looking. I used to see these in the hippocampus quite a bit. And actually they're positive for C1q complement. And these are thought to reflect a more chronic inflammatory state. And so the morphologies are pretty clear that in terms of you can distinguish these. And so that those counts were done. And it was absolutely interesting what came of this. It was completely unexpected. So when you just trying to represent these data, it was also a little bit of a challenge. So if we just look at the gray matter or frontal cortex of all these groups of cases, and we look at just normal aging across the top, young, middle age, and age controls, and the proportion of these different microglial phenotypes, what you can see is with aging, the big blue is dystrophic. Not surprisingly, we see more dystrophic neurites with age. I'm sorry, dystrophic is the dying ones. We see more dying microglia with age. We also see the hypertrophic, which are the ones that are responding to injury in the gray pie slot here, increasing with age. And the number of ramified or quote resting surveillant microglia in the white decreases with age. So that's what you might expect for normal aging. So now when we look at Alzheimer's disease, just focusing on here, we're seeing far more dystrophic microglia, lower numbers of resting microglia, and not surprisingly, we see lots of hypertrophic microglia. So here's what surprised us in Down syndrome. When we looked at younger Down syndrome cases without AD pathology, they looked very similar to young controls. So that was that was fine, kind of what we expected. But when we looked at Down syndrome with Alzheimer's disease, we saw a huge number of dystrophic microglia, larger numbers actually compared to sporadic AD. And we lost a lot of those ramified, but we don't see as many hypertrophic microglia as what we see in Alzheimer's disease. And so I'll show this in a different way. When we plot the individual data points, which I think is super important to do to see if there's somebody driving this effect. Here's, we're just looking at ramified microglia, so those resting microglia. We don't see a lot of these in white matter, but we do see a lot of these in gray matter. And we compare just young controls to Down syndrome and middle-aged controls to Down syndrome with AD. We're seeing, not surprisingly, a decrease in the number of these resting microglia in Down syndrome with Alzheimer's disease brains. So that's good news. In other words, it's similar to what we see in sporadic AD. However, what was really interesting, again, is we are seeing far more dystrophic microglia in the Down syndrome brain. And so that might give us a hint that the microglia in Down syndrome brain are either under a bigger load in terms of trying to clear up pathology, or they're unhealthy compared to what we see in normal aging. And then those microglia are really struggling to do their job and are dying as a consequence of that. And maybe that's a pathway we really need to look carefully at in Down syndrome, given these different neuroinflammation phenotypes. And again, that has implications for clinical trials in future. But surprising to me was the lack of really big increases in hypertrophic microglia. And I think that's because that phenotype jumps very quickly to dystrophic. And we're not seeing a lot of these busy, active, taking up stuff and dealing with damage that we would see in the hypertrophic microglia. And then when Ali and Alex published this study, I was so happy to see that another group independently essentially was doing the same study in another cohort of brains of people with Down syndrome. In Claude Coelho's lab in Montreal, and Lici Flores Aguilar did this gorgeous study looking at across the lifespan and mapped down a number of pathways. But what she was also seeing here, and I'm just going to highlight, is these changes in the morphology. But she had a beautiful way to kind of quantitate that hopefully she will teach us, because she's now joining us for a postdoc and starting now, as soon as COVID lets her come in. So there's really something to the story. And it's, I don't know what the molecular mechanisms behind all of this are yet, but I'm hoping Lici will take this on and continue to work on it. And it's not only just important to think about these neuroinflammation markers in brain and thinking about clinical trials and treatment targets, but these neuroinflammation markers also have a biomarker role. So Melissa Peterson, who's a junior faculty member at University of North Texas, has been looking at the plasma of people with Down syndrome in our ABCDS study, and measuring and doing a proteomics kind of profile of protein changes that happen when we look at people who are cognitively stable with Down syndrome relative to those who are developing mild cognitive impairment, or the best that we can assess that in people with Down syndrome. and compare those to people who are frankly have dementia and also have Down syndrome. And what Melissa found in this kind of examination is that when she compares people who have dementia with Down syndrome to those who are cognitively stable, the biggest hits she sees in terms of which proteins in the plasma distinguish those two groups are inflammation. A lot of inflammatory molecules are engaged here. And similarly, when she looks at mild cognitive impairment in people with Down syndrome and compare those to people who are cognitively stable, again, she sees a lot of inflammatory proteins kind of distinguishing those two groups, plus some vascular proteins, which is a great lead for us. So this story about neuroinflammation and inflammation in people with Down syndrome is a big deal, I think. And not only is it important in childhood, and that's a whole different discussion, but certainly it's happening with age, it's happening with Alzheimer's disease. And I think to some extent that pattern's a little bit different than what we see in sporadic AD. Very important for clinical trials. So let's start kind of putting this all together, and I'll bring this kind of to a close. So the way I've been kind of conceptualizing this is to put all of these neuropath pathways into an aging axis type of format. So here's the original image I showed you where we're seeing frank AD pathology by 40, signs of dementia in their 50s. But if we start kind of slotting in all these other pathologies I've just told you about, we can see that there's some early neuroinflammation that happens before Alzheimer's disease. And I left off this part of the story entirely. We start to see cerebral amyloid angiopathy and microbleeds in the 40s and 50s. And then we see a second kind of burst of a different profile of neuroinflammation, and then higher prevalence of these dying microglia. So again, thinking about treatments and orders and maybe what leads to what, this is kind of a really fun thing to do when we study Down syndrome to understand Alzheimer's disease that we can't necessarily do easily in the general, in sporadic or late onset Alzheimer's disease. And indeed, kind of what the whole lab is focused on, everybody's contributing a piece to this project in the lab, is to put this in context of the Jack curve, which I'm sure you're all familiar with, which is typically applied for biomarkers. But I'd love to apply this in the context of neuropathology. And so Ira and I put our heads together and kind of started this conceptually to think about, what ages do we start to see these different pathways kind of engage? And we're going to try and now fill these in about where does the vascular pathology come in, and maybe the different kinds, and when does the inflammation come in and what kind, and try to fill out this whole Jack curve in the context of neuropath. So what are we working on now? We got some really fun projects going on, and I'm just putting a picture drawn by another graduate student here at UCI. Her name is Batul Rizvi. She's an incredible artist, and so I use her artwork everywhere I can with permission. And so she does this beautiful artwork in neuroscience. And we have a couple of really fun projects that we're trying to start really dig into this vascular story and inflammation story and try to put these stories all together, because we're using the same set of autopsy cases for all of our work. So we can talk about vascular pathology and other pathologies all in the same set of cases, all on that x-axis that's going to be the same. So Lorena Sordo just joined us as a postdoc from Scotland, where Lorena actually did a huge amount of work on the cat model of Alzheimer's disease. Beautiful work. So she's starting to look at some of the vascular pathology. And of course, this has been a really, really exciting area of research in the literature. Lots of great people looking at this. And so taking a lot of hints from these folks. But one of the areas we're looking at is extravasation of fibrinogen, because this has implications for neuron function in the proximity of fibrinogen getting into the brain. And these are some pilot studies we just did for our grant. So you can these are like single or 2 case studies, but I think they're really intriguing. And what Lorena saw was there was clearly in areas where there were blood vessels, you can see them here, where the spaces are, in Down syndrome brain with Alzheimer's disease, there's clear fibrinogen leaking out into the brain. So those blood vessels are not doing good. And then we did a study of just anti-collagen, just for large blood vessels in the brains of people with Alzheimer's and with Down syndrome, Alzheimer's disease, and sporadic Alzheimer's and old controls. And really, we just wanted to do this because we wanted to set the stage for doing double labels. But when we looked at the slides, we were really struck in that you can see these beautiful big blood vessels in Alzheimer's and non-demented elderly controlled brains. But in Down syndrome, those blood vessels don't look the same. So we don't know what this is yet. So this is something we're gonna have a lot of fun playing around with to see what this means. And of course, I've been talking a lot about microglia, but the other big glia cells that we need to think about are astrocytes. And given that S100 beta is overexpressed in Down syndrome, astrocytes have got to be a part of this story. And so Leslie Vasquez-Rangel, as an undergraduate student, is doing gorgeous work looking at astrocytes, labeling them with different markers to give us an idea about their function. And she's doing a lot of immunostaining and imaris quantitation to characterize these astrocytes in gray and white matter. And then Shiley-Marcos is another graduate student in the lab, and Shiley has landed on a protein, aquaporin 4, which is really fascinating. Aquaporin 4 kind of links the flow of fluids from the vasculature into the brain. And there's some work suggesting this protein receptor is mislocalized in Alzheimer's disease, and we have no idea if that same thing is happening in Down syndrome. So she's going to be hitting up the super resolution folks here to get a handle on that. So I'm really excited about both of their projects, and they've done a lot of work to figure this out on their own. So let's kind of pull this all together, and then I'll finish up with a little bit about ABCDS and give time for questions. But I hope I convinced you that Down syndrome is associated with early onset Alzheimer's disease pathology, that amyloid precursor protein overexpression and cleavage to beta-amyloid is probably what's leading to more cerebral amyloid angiopathy. Certainly, when we see more CAA, and there's another paper I didn't tell you about, but Alex clearly linked the CAA to the microbleeds. So we think that CAA is leading to leaky blood vessels and or brittle blood vessels that are breaking, and blood is getting into the brain. And that blood getting into the brain is bringing in serum proteins, maybe causing a different pattern of neuroinflammation in Down syndrome, or it certainly is more prominent in Down syndrome brain. And it's thought to be linked to the formation of immune complexes in the brain stimulating microglia. And when we looked at microglia, we see fewer resting microglia, which is not unexpected, but what was unexpected, which we're seeing a lot more dying microglia in the Down syndrome brain. And these are linked to, plasma biomarkers as well, which is really fun to think about. And we're going to keep running down those pathways of looking at vascular pathology and the neurovascular unit and the astrocyte story. So I'm hoping if we get together again in the future, I'll have some more updates on that. So what are the implications of some of this work? And I think really that there are all multiple targets for intervention for people with Down syndrome that might target A beta, might target tau, might target cerebrovascular pathology and neuroinflammation. Maybe we have to consider, and not surprisingly, multiple prong types of approaches. However, because we can understand these different events in the context of aging in people with Down syndrome, we might be able to get a little bit more precise on, based on the age of a person with Down syndrome, what the best targets for intervention would be for that person. And maybe, given the timelines of how aging and Alzheimer's disease develop in Down syndrome, they might be one of the early cohorts that really benefits from a prevention study, since we kind of know the age of onset of each of these pathologies. But we can't really implement these clinical trials and prevention studies super duper well yet, because we still need to figure out what the best clinical outcome measures are for doing these intervention studies. We also need to understand it just in terms of aging. And because I think in future, it's going to be very important to have clinical trials that are specifically dedicated to people with Down syndrome that takes into account their unique features. It does break my heart that almost all clinical trials for Alzheimer's disease that are running right now specifically exclude people with Down syndrome. And I think we need to change our attitude on that and be more inclusive of people with Down syndrome into these clinical trials. So one of the big efforts to kind of get us to a point of clinical trials is the Alzheimer's Biomarker Consortium for Down syndrome. And I put the web link up there. Pretty much if you Google ABCDS NAA, you'll find our website, where we're following 550 people with Down syndrome over the age of 25 years. We have 18 clinical and research sites. It's a pretty large endeavor. And in this study, we bring people in every 16 months for a clinical examination, for a neuropsychological exam. And we pull blood, and also, if people are willing, we also collect CSF samples. There's extensive neuroimaging in terms of PET, tau, and amyloid, and also MR, lots of outcome measures with MRI. And of those people who consent to brain donation, and we're trying to encourage people to think about this a bit more, we'll follow that up with neuropathology data when the time comes. So these sites are all around the country. We also have a site in the United Kingdom, and I'm hoping in the next iteration of the ABCDS, we'll have more sites both internationally and nationally. But this is really an exciting and fun study, and all of the data, imaging data, proteomics data, metabolomics data, genetics data, are all deposited into data warehouses that are accessible by anybody. You just put in a request and we can share that data with you. So we're encouraging people to use this data as much as possible. So I want to put out a big thanks to the NIH for supporting all of this research, and also the Bright Focus Foundation, which has been helping us with our brain banks. And so thank you to them. Again, many thanks to our families and our participants. And I also want to thank my lab, which is pretty fantastic. We have a great time together. And of course, when your mascot's a dog, that should tell you a lot. But our lab is made up of postdocs, graduate students, undergraduate students, and high school students. So we're pretty inclusive of folks who are interested in looking at this in more detail with us. So with that, I will end.